as kind of a summary of how do molecules move around in the body. All right, so first one is diffusion. If you have a difference in concentration and a path for the molecule of interest to move, it will move from an area of high concentration to an area of low, right? That's diffusion. That's that free movement because of Brownian motion. All right, so that's A. In C here, this is our hydrostatic pressure movement. This is how the heart moves fluid around the body, okay? The heart contracts. By contracting, it lowers its volume and increases its pressure until the pressure inside the heart is greater than outside the heart, and therefore blood gets pushed out of the heart and into the circulation. We'll talk much more about that in a week or two, all right? And then we get down here. Some molecules will move simply by their charge, okay? So in our example over here, we have a lot of negative charge on the outside and less negative charge on the inside. So we have a positive on the inside. Well, that means molecules are actually pushed from the negative outside to the positive inside. All right, we're going to talk about this when we go to build membrane potential, right? Because molecules are not only pushed by their concentration gradient, they're pulled and pushed by their charge difference too. All right, so more on that in a minute. And then this last one is an example of osmosis. Remember, this is how do molecules move? Well, one of the ways water moves is towards areas of high concentration. The primary reason that's true is, except for two small exceptions in the body, all human cells have channels for water to pass through. Okay, those channels are called aquaporins, and they're present in all except two places, a tiny little place in the brain and one particular part of the nephron in the kidney. Otherwise, cells are permeable to water, but they're not permeable to everything else. So we have a semi-permeable membrane, right? Cell membranes can be considered semi-permeable membranes. So if you have a high concentration of solute inside and a low concentration of solute outside, water is going to be pushed in or pulled in by the high concentration, and we'll get net movement of water that way. So like in this example right here, we got a lot of sodium inside, not very much sodium outside. This cell would swell, right? It would get larger in volume because more and more water would come in until it was so full that water couldn't enter anymore because of the pressure, right? A little more on that when we get back to osmosis later on in the semester. Okay. All right, so if we look a little bit at types of active transport and co-transport, we have some important examples here. Okay, so this is called the sodium glucose co-transporter, and it's an excellent example of what we call co-transport. All right, so if you're going to move a molecule up its concentration gradient, right, from low to high, you're going to have to put in energy somehow. All right, the classic way of doing that is just burn up an ATP, right? That's easy. But our cells are very efficient, so they don't want to burn ATP if they don't have to. So sometimes what our cells will do is this little trick where the cell will move one thing up its concentration gradient using the power of something else moving down. Okay, so you see sodium is going from outside the cell to inside the cell, right? That's down its concentration gradient. There's an energy there, okay? Well, as the sodium moves down, it makes this protein change its shape so that the glucose is sort of pushed down into the cell as well. Now, this is sort of a double whammy because glucose is also moving down its concentration gradient, right? So you have two molecules moving, and the glucose is sped up by the sodium falling down its concentration gradient, moving those molecules around, right? So this one is driven by the sodium concentration gradient. And what it means is using the sodium glucose cotransporter, we can actually move glucose from an area of low to an area of high concentration. We can lift it up, its concentration gradient. So like imagine in the liver, for example, when you're just post having eaten a high carbohydrate meal. So the liver cells have lots of glucose in them anyway, but we need them to take in even more so that they'll store it up as glycogen. This sodium glucose cotransporter makes that happen. What I'm trying to point out here is there's no ATP in this picture, right? But energy is still being used because sodium, it only got to be high concentration to low concentration because of the sodium potassium pump, which did use an energy. Do you see? So we'll talk about this a couple of more times, but it's an added efficiency. Think about it like this. You know, there's power plants that generate electricity from falling water, right? Well, what we have here is we've essentially we moved the water for free, and that falling water is then generating more energy to move another bit of water, right? So more on that analogy later on. Okay, this side, perhaps the most important of the cellular pumps, period, bar none, for real. Here we are, second week, but this one. The sodium potassium pump is a highly refined pump that moves sodium out of the cell and potassium in the cell. If you look back at that slide that shows you the differences in concentration inside and outside, this is the reason why sodium is high outside and potassium is high inside, is because of the sodium potassium pump. Now, why can I say that so strongly? All cells leak sodium and potassium. Okay, in other words, if you take a cell, a dead cell with no energy, and you put it in normal blood, the sodium will leak in and the potassium will leak out. All right, they are not ion tight, so to speak. So the only way we maintain this difference, high sodium outside, high potassium inside, is we have to use ATP to pump it around. This pump is hugely efficient. It moves 3 sodiums out and 2 potassiums in for every ATP that's spent. So it's highly efficient. Because it's efficient and because it's working all the time, okay, this sodium-potassium gradient is used to power other things, right? So here, we're taking sodium and evicting it from the cell. That's where we spent our energy. Over here, the sodium's falling back into the cell with such energy that it's able to move something else. Do you see? So like the sodium glucose pump is actually powered by the sodium potassium pump because this pump created the concentration difference in the first place, right? We're going to see a lot of that. The body uses that mechanism, moves sodium to move something else quite a lot, all right? So we're going to see that in the GI tract. We're going to see it in the renal system. We're going to see it quite often. Okay, so that's enough. This is a classic active transport. Okay, then here what we have are what we would call co-transporters. We'll just call them co-transporters. Here, first co-transporter, they're both moving in the same direction. These two show you that doesn't have to be the case. You can have counter-current co-transport where the two things are going opposite. So for example, here, sodium falling down its concentration gradient, which was created by this in the first place, right, is pumping calcium out of the cell. Remember, I made a big deal about how calcium inside and outside very, very different, right? Well, how did it get to be that way? In part because of pumps like this, co-transporters probably.